Welcome everybody to this Wednesday afternoon where it's apparently stopped snowing for now, at least at the west end of the city here. Um, we are fortunate today to have Dr. Ryan Stevenson with us to give a presentation. And I'd like to believe I'm a reasonably honest person. So when I say fortunate, I'm not saying that just to be polite, that Ryan actually has done some great research and is also a great guy. So combination of two important things. So Dr. Stevenson completed his doctoral work at Indiana University with Bobby Knight.
Oh yeah,
All right, you and him were tight.
threw chairs together, everything.
Excellent. But had previously done his bachelor's degree at the University of Michigan. He then was an NIH-funded postdoctoral researcher at Vanderbilt, and then did a Banting Fellow at the University of Toronto, all in the area of sensory phenotypes and processes. Correct.
So,
So a lot of really specialized work in this area, and in autism as well, and is now a member of the Department of Psychology at Western, where he is also the associate chair for research, a Killam fellow, sorry, and a member of the Center for Brain and Mind and the Western Institute for Neuroscience. So a lot of appointments, I think, reflecting a lot of the work that Ryan has done and a lot of the publications. So today, Dr. Stevenson will be talking about sensory phenotypes in autism from neurobiology to clinical associations. And I'm looking forward to an excellent talk. Ryan, so the floor is yours.
all right, thanks, Rob. I'm actually even since then I had one of my grad students was I was just telling this to Jason was still sending me figures to add in till about half an hour ago. So I actually have some ADHD stuff in here too, if we have time to
Great.
get to
Yep.
it. Okay, so I know with the audience here, most of you will be very familiar with autism, but just a quick overview. So autism is a neurodevelopmental condition with complex etiology. We know there's a strong genetic component, but there's a ton that we don't know about it. It has quite a high prevalence rate. We're up to 1 in 36 births, according to the CDC. And with the new, well, not new anymore, the 2013 DSM, it's characterized by two clusters of affected domains, social interactions and communication, and then restricted and interest and repetitive behaviors. Also, with the new DSM, they finally included atypical sensory processing as an actual diagnostic component of autism, even though they've known since some of the earliest depictions in the 30s and 40s that atypical sensory processing was at issue. And one of the things that I think has really come to the fore in the last decade or so is that these sensory issues really impact what are like sort of the old standard issues with social communication and restricted interest and repetitive behaviors. If you talk to autistic self-advocates and things like that, they often will say that things like stimming are done to actually alleviate sensory issues that they have, you know, the overwhelming sensory environment, and things like that. Obviously, when you're talking about something like speech communication, you have to be able to it's completely based on your ability to perceive what's being said and the movement of voices and facial expressions and things like that. And this is while it's not necessarily the most pressing issue for many people in autism, it is perhaps the most common complaint in autism. Up to 94% of individuals are impacted. And this goes across sensory modalities. It can be in vestibular, vision, audition, olfactory, everything. So one of the reasons that I got really interested in looking at sensory processing specifically was that sensory processing is really this very low level building block that I would argue pretty much all cognitive processing is built upon. So if you think of your whatever your favorite cognitive processing is, emotion recognition, memory, things like that, all of these things require proper sensory processing to really occur. So if you think of memory, for example, if you see and hear something differently than I do, it's not a huge logical leap to think that you'll remember it differently, right? Same with like social communication. If you perceive facial expressions differently than I do, then you're probably going to learn how to interact with different emotions in a very different way, which highlights the second bullet up here is that there's also a developmental factor. So it's not just an individual um, moment in time. The development of most of your cognitive processing is also based on how you see and hear and sense the world around you. So your memory systems, your emotional systems are going to develop differently if the information that you're getting from the outside world is processed in a different way than your peers around you. So this can make, to me, it can make sensory processing a very important component of autism. One of the issues though that we run into, especially in terms of like dealing with sensory issues, is that they're extremely heterogeneous. I mean, there's the standard cliche: if you've seen one kid with autism, you've seen one kid with autism. So yeah, so this can make things like developing and delivering supports really difficult, because if everybody's unique in the way that they present their sensory issues, then you're going to have quite individualized support systems for everybody. Um, but one of the things, what I want to talk about today is whether or not that heterogeneity isn't quite as heterogeneous as we think it is. So the overall sort of base of this talk is to look at whether or not this heterogeneity is random, if it's actually truly unique across individuals, or whether there's some patterns to it, some subgroups that we can look at. And these are what I'm going to call sensory phenotypes. So phenotypes where across kids, there are similar patterns of which sensory issues sort of cluster and go together. So that's going to be the first of four research questions that I want to talk about today. So first, are there stable, meaningful sensory phenotypes in autism? This is a 50-minute talk, so you can probably figure that I'm going to say that there are, but I'll show you what they are. We wanted to look at whether or not these different phenotypes are clinically meaningful. Do they have different clinical presentations or different demographics that go along with them? We also wanted to get into whether or not they have unique neural signatures. Because a lot of things like what Rob is doing in terms of like looking at pharmacological interventions and things like that, you want to know what's going on in the brain with these. And last, if we have time to get to it, we're going to look at whether or not these phenotypes are specific to autism or whether or not they're transdiagnostic across different neurodevelopmental conditions. So all the data that I'm going to be presenting to you today comes from the PAWN network, which both Dr. Nicholson and I are a part of. So we've recruited so far over 3,000 participants for these. This data has been associated with over 100 publications, 50 investigators. It's run out of, I'm going to miss some of these, but Sick Kids, Holland Bloorview, Queens, LHSC, Western, McMaster's, and a number of other research institutes. So in this network, the way that they've measured sensory processing is through a really simple measure. So it's called the short sensory profile. It's a simple pen and paper, or it's online now, but a survey measure where parents will report what their kids' sensory issues are. So what they measure are 7 different subdomains. They measure tactile issue, taste and spell, movement issues, under responsivity and sensation seeking, auditory filtering, low energy and weakness, and visual and auditory processing. So these are the 7 different measures that you get from the short sensory profile. And so I had this student, Christina Curson, was actually an undergraduate honors student who came up with this idea. She was reading a few papers and was like, hey, do you mind if I do my honors thesis on looking at sensory phenotypes? And this was maybe about 6 years ago. And now we've got like a number of papers out on this. We have a CIHR grant all based on this undergrad's idea, which is really cool. And then it was later carried up on by a postdoc of mine who's now a faculty member at Laurier, Nikki Shearer. So what Christina wanted to do is she wanted to take these 7 different domains and see if they were randomly distributed across all of these autistic participants, or whether they clustered together in meaningful ways. So to do this, she used a statistical technique called a k-means cluster analysis, which I do not expect any of you to have heard of. But essentially what it does, if you think of 2 different variables, so here they're labeled x1 and x2 on the x and y axes respectively. If you just take all of your data and you plot it out, it looks like a huge cloud of dots, right? What k-means cluster analyses does is it looks for patterns, it looks for clusters within these dots. So you may get a solution that looks something like this. where you have on the y, whatever the measure is on the x-axis, if you're low on that measure, but also low on the y-axis, you get this red cluster, right? And the main idea here is that these individuals on these two different measures are more similar to each other than the averages are for this cluster, the red cluster, and say the green cluster. So this is in 2 dimensions. We have 7 outcome variables. So this is in 7-dimensional space, so I can't actually show it to you. I'm not a computer. But so what this does is it does this clustering in 7 dimensions and looks for patterns across it. The way we chose to do this was to force it to make a certain number of clusters, and we slowly progressed and added clusters until the different sensory phenotypes lost clinical meaningfulness, and when we stopped accounting for new variability. So what those look like, if I can take you up here, what this looks like is on the y-axis here, you have whether or not they're better performers in terms of sensory processing, which is up, or worse performers relative to this group's mean. Each one of these bars indicates a different aspect of sensory processing. So we've got tactile, the purple bar here is taste and smell, the aqua bar is movement, and so on and so forth. So when we force it to cluster all of our autistic kiddos into 2 clusters, it has an outcome where it has one group that's above average on all sensory processing and one group that's below average on sensory processing. That's great. It's kind of boring. It's like, okay, so these are the kids that are more severely impacted. These are the kids that are less severely impacted. So then we progressed along and looked at a 3-cluster solution, a 4-cluster solution, and so on. As we progressed, these bars indicate the thickness of the bars indicated how many individuals sort of stayed within this cluster versus went into the new clusters that were extracted. So you can see here in this 3-cluster solution, it's still sort of that, we have the high achieving kids, the sort of average kiddos, and then the kids that are really having a lot of sensory issues. Again, not super interesting. But as you move on into the different solutions, as you add solutions, you can really see things start to emerge when we get to a 4-cluster solution, where we still have this high-performing group and a low-performing group. But now you're seeing, for example, this cluster of kids, which represents 25% of the group, that really just has issues with taste and smell. They don't have issues in other domains. Or this group who has issues specifically in auditory filtering here and under responsivity and sensation seeking, but tend to do fairly average or above average in the other domains. So this is really what we're looking for in terms of sensory phenotypes. The last one we did was a 5-cluster solution. So here we still have our taste and smell, our under responsivity folks, but we ended up having a group that also had issues with low energy and weakness. So this is sort of like a vestibular, muscular issues. This ended up being our optimal solution, which left us with 5 clusters that I'm going to go over in more detail. Don't worry about those. You'll see them right here. So the first we're going to look at, so we had to label these. So the first we labeled was a sensory adaptive phenotype, which accounts for about 1/4 of the autistic kids. These were kids that across the board were above average in terms of sensory processing. And we had a second group that we called generalized sensory differences. These are kids that just sort of struggled in terms of sensory processing across the board. The last 3, to me, are actually the more interesting ones. We had one group which had issues with under responsivity and sensory seeking, and I'm labeling these based on the issues where they have the most issues with. So they do have positives too, like they have no issues or they do much better in taste and smell, but we're gonna label them based on where we need to help them and provide supports. So this group, had issues with under responsivity and sensation seeking. The third group was that group that had taste and smell sensitivity in particular. And a fourth group that had issues with movement and low energy, particularly the low energy and weakness scale here. So in terms of our first question, yes, autism does cluster into meaningful sensory phenotypes, but we really wanted to see of particular interest was whether or not they were clinically meaningful. It's great if they cluster together, but is it something that actually impacts them clinically? So I actually had a little poll that I was going to do, but I didn't realize that I wouldn't be able to see all of your faces and see you raise your hands. But so maybe just do this in your head. But the first thing we wanted to look at was whether or not they had differences in cognitive abilities. So again, thinking of that we have these five different phenotypes, just form a hypothesis in your head about which one you think would have like lower general cognitive abilities. You can see if you felt the same as we did. So my first thought was that the group with generalized sensory differences, the ones that were more affected in terms of sensory processing, would probably just across the board um, be more impacted and would have lower cognitive abilities. Turns out we were very wrong. There actually were no significant differences across cognitive abilities in these groups. And in fact, and numerically, not statistically, but numerically, the folks with generalized sensory differences actually had higher IQs than those um, that had the better sensory processing. So no differences in cognitive ability. We also wanted to look at demographics across these sensory phenotypes. So we looked at age and sex in particular. We did see significant differences in age, specifically that the kids with taste and smell sensitivities tended to be significantly younger than all of the other groups, and those that were more impaired on low energy and weakness, or described as low energy and weakness, were older. Now, I should emphasize that this is not a longitudinal study at this, or this data is not longitudinal anyways. But one thing that would result in something like this would be that you actually see kids kind of growing out of one sensory phenotype and changing into another. So in this case, you could hypothesize that those kids that had taste and smell sensitivities sort of outgrew them and ended up moving into one of these other phenotypes, probably specifically this low energy weakness one. And just going from parent reports, if you think about the eating issues that kids have, particularly when they're younger, in autism, that could be what's leading to this lower age for the taste and smell sensitivity group. In terms of sex, there were no significant differences in terms of sex. So the male, female, they were all significantly male weighted, as autism is in general. We also wanted to look at the other core autism traits. So we wanted to look at social ability. So we measured that with the social communication questionnaire. And here you see, and repetitive behavior, so the other side of autism, you see sort of a similar pattern across these different groups, where the sensory adaptive groups are less impacted. So up here is higher trait severity. The generalized sensory differences are the worst performing, and then we have these two groups, the under responsivity and taste and smell folks doing a little bit better than the low energy and weakness. And these two actually were not significantly different from each other. In the social abilities, they were significantly different from each other here. So this is kind of showing us that, at least in terms of autism, issues across the board, that there were clinical differences in how they were presenting. But we also wanted to know, I mean, a lot has been coming out about how folks with autism often have secondary diagnoses or three diagnoses, right? So we wanted to look at other really commonly co-occurring symptoms. So the first one we turned to was ADHD. So we measured inattention and hyperactivity using the SWAN. And here we see a different pattern. So we see the folks with generalized sensory differences and the under-responsivity sensation seeking were significantly worse in terms of inattention than all of the other groups, particularly the low energy and weakness and the sensory adaptive group. And when you get to hyperactivity, this under-responsivity sensation seeking group actually takes the lead, so to speak, and is showing the most impairment here. And this is something that's going to come up a little bit later in the talk, but looking at these data and this is the group, they had under-responsivity, sensation seeking, and auditory filtering issues, which to me, I'm like, oh, I wonder if these kids, so these kids only had ASD. We did not include them if they had ADHD. So they don't have ADHD, but to me, it looks like they're showing a lot of tendencies for ADHD, which was sort of validated by this. But the other really cool thing to note is that these clinical profiles don't actually line up with what we see in terms of the social abilities and the restricted interests and repetitive behaviors. They're not ordered in the same way. So you're actually seeing it's not just a level of severity across the board. These sensory phenotypes are associated with specific clinical issues. We also looked at OCD. And here we saw another different pattern where the sensory adaptive, so this scale happens to work in the opposite direction. So the less negative you are, the lower, the more impaired you are. I don't know why they scored it that way, but that's how they score it. So here we've got the sensory adaptive show the least number of OCD traits. These three groups were all not significantly different from each other, and we saw the most OCD traits in the generalized sensory differences group. So as a little bit of a mini summary, I'm actually doing okay for time now, which is great. So we were able to show that sensory issues in autism cluster into these predictable phenotypes, that it's not necessarily an individual unique presentation, but that these traits actually cluster together. And importantly, that these different phenotypes exhibit different clinical profiles. So there is an actual clinical relevance to them. We also wanted to see whether or not there were different neurobiological substrates. So for those folks who are doing things like looking for targeted pharmacological interventions and therapeutics and things like that, like where in the brain should they be targeting if we know what phenotype these kids show. So this was done by two postdocs of mine. Now Professor Kathleen Lyons, she's at King's University now, and Dr. Eunjung Choi, who works with us at Western still. So we started out looking at neural differences in terms of functional connectivity. So we have had about 400 kids who had good functional scans and also had the sensory profile, so we could see which phenotype they were in. And what we're going to do here is we're going to do an analysis looking at neural connectivity. So how strongly related one brain region's pattern of activation was to another. So we started and we parcellated the brain using the standard method. I'm not going to get too much into the weeds here, into 400 different functional areas. And then these functional areas are collapsed into 7 different networks that are very commonly found and we know sort of fire together. A visual network, a somatomotor network, the dorsal and ventral attention networks, limbic networks, frontoparietal networks, and a default mode network. So you can see all those are color-coded on these. And this measure that we're going to take, what we're going to do is we're going to extract so the kids are sitting in the scanner. They're either not doing anything or they're effectively watching what looks like a screensaver. So it's just how their brains naturally not doing a task fire, right? So we'll measure the activity from one area here, and you might get something looking like that. And we'll measure the activity from a second region. And what we're going to look at is we're going to see how tightly coupled this brain activation is. So is this area in the network, in the left dorsal attention network, does it synchronize with the same area on the right side of the dorsal attention network? And we're going to look at all of these. So we're going to get a correlation value for these two patterns. And those we're going to plot out into this huge matrix of correlation. So each little point on here is a correlation value, an R value, between 2 different regions. What's blocked off in these black areas here are the networks that I had identified before. So anything inside these boxes are within network patterns, and anything outside of these boxes are between network synchronies. So you can see that within network, you see a lot more synchronization. So if you like, which makes complete sense, right? So in the visual domain, the voxels in the visual region are way more likely to show a similar pattern of activity to each other than, say, the visual areas and the limbic system, right? Those are going to be processing different things, so they're not going to be quite as correlated. So this is in our non-autistic sample, right? So then what we did is we wanted to look at these patterns in our sensory phenotypes and do a couple things. One is, were they different than were the autistic and non-autistic participants different? Did they show different patterns of neural connectivity? But then more relevant to the talk today is, did the different sensory phenotypes actually show differences? So this is what I'm going to present is kind of a very broad strokes, right? We're only looking at 7 different networks and the difference between within network connectivity and between network connectivity. But we'll get into more detail. So we take these matrices that we have here, and we calculate them first for each individual group. So here it's really hard. So I'm just showing you these. Don't try and find the differences yet. I'm going to directly compare them. But you can sort of see a little bit, like a little bit less between network connectivity here. And you do that for all of them. So again, it's hard to really compare these because they're so complex. So what we're going to do now is I'm going to go through and I'm going to directly compare each one of these to the non-autistic and just do a subtraction. Actually, that's not true. The first thing I'm going to do is I'm going to collapse across these and I'm going to show you that comparison between autistic and non-autistic. What it's going to look like is it's going to change from just these correlations to, it's going to change to T values. So it's going to be red and blue. Red is going to indicate greater connectivity in the autism group. Blue is going to indicate greater connectivity in the non-autistic group. And what we get is something that looks like this. So what you see is in the autism group, you see a lot more within network connectivity, but significantly less connectivity between different networks. And you can see this is where these differences were, the significant differences were distributed throughout the brain. They're all throughout the brain. It's a very, very generalized difference. So this was not surprising at all. This has been shown before, but it's a bit of a sanity check that we can see these differences between within and between network connectivity. But again, we wanted to see whether these patterns differed. So do we get this exact same pattern across different phenotypes, or do the different phenotypes look different? So our first comparison that we're going to do is the typically developed kiddos versus the sensory adaptive. So again, these were the autistic kids that were fairly good with all of the across the board in terms of sensory modalities. And you just see sort of across the board, you see differences. You see greater connectivity in the sensory adaptive group. Again, particularly along this diagonal is where those within network ones were, right? And then you look at the generalized sensory differences and you see a very different pattern. So it was hard to tell the difference when you put just the two next to each other, but when you plot out the differences relative to the non-autistic group, you can really see that this group is the sensory adaptive has a lot of sensory over connectedness, and the generalized sensory difference is showing a very different pattern where they're showing under connectivity. The one exception you'll see this T, it's going to pop out here and here. This is the limbic system. The limbic system, especially for this generalized sensitivity group, looks very different than everybody else. If you look at that sort of ADHD-like group, the under responsivity and sensation seeking group, you see lots of overconnectivity. Again, especially within networks. So you look at the default mode network here, and you can see that the default mode network is just really, really highly connected in the autism group. Same with the somatomotor group, or with this specific autism group. That is spelled wrong. That's a typo. Sorry. It should be URSS. And then we get to this is the low energy and weakness and the taste and smell sensitivity. Again, the big thing that I want to highlight aren't specific regions here. I just want to show that there are very clear differences in neural connectivity across these different phenotypes. You can also see this if you do the direct comparisons between these groups. So like sensory adaptive versus the generalized sensory differences, you see those same patterns and you just get very drastic differences, right? So you can see the comparisons between sensory adaptive and generalized sensory differences versus, say, the generalized sensory differences versus the under responsivity sensation seeking. So you can see, I mean, they're quite clear differences. And here again with this generalized sensory differences, you see like the limbic system here just popping out a ton. So the big take-home message from this category is that yes, sensory phenotypes definitely clearly have different underlying neurobiology in terms of functional activity in the brain. But again, this lets you look at like really broad stroke networks. It's good to have a more detailed look at where these differences are. So for this, we had a smaller group that had good, really high quality scans, but also had the sensory profile conducted. So there's between 20 and 40 in each group. And for this, what we were going to do is we were going to use a machine learning algorithm to see if we can take the brain information and predict which phenotype each kid was. So we're sort of going in the other direction. We wanted to see not just where in the brain, but we also wanted to see what sort of functional attributes these connectivity patterns here. So to use that, or to do that, we used a couple different graph theory measures. Again, I don't expect anybody to be an expert in graph theory. I'm not the guy who did this is. So I'm going to just do a really quick blitz through what these four different graph theory measures that we used were. And so this was work done from a PhD student at Western, Matthew Koliznik. So we're going to use four different measures. The first two here are going to measure sort of how strongly connected a network is. So the first is called local efficiency. So efficiency is basically if you knock one node of the network out, how much does it disrupt everything? So you can see in this case, if you knock out this red node, everything will still be okay because it's connected. If you knock it out on this side, the whole network falls apart because these four nodes aren't connected to each other. So that's what we mean by efficiency, is how sort of robust it is to damage, I guess, or robust it is to a node falling out. The second measure that's highly related to that is called the clustering coefficient. So it's how tightly connected a network is. So a clustering coefficient of 1 means every single node is connected to every other node. A clustering coefficient of 0 means like for this particular node, this node that it's connected to is not connected to any of the other nodes that this purple one is connected to. So those two are quite related to each other. The other two are measures of what we call centrality. So how critical a node is to a network. So in this case, if you knocked out this node, it has a really high between the centrality measure. It is the only connector between these two networks. So this is a really important node, and it's sort of central to the way that this operates. And you can also look at this using node strength. So how many connections within a network does a node have? So if you knocked this node out, a lot of this network would become disconnected also. So you don't need to, those are just what we're measuring. So you know what we're doing. You don't have to memorize those or anything like that. So what we did is for each voxel in the brain, we took those four measures and we ran some machine learning algorithms to try and use that information to classify each individual to one of the 5 sensory phenotypes. So we did that using a technique called a linear support vector classifier, which sounds really, it's basically a regression. So if you just know a regression, you have the basic knowledge to figure out how to do this. So what it does is you have two groups of people, not two groups of people. You do have two, in this case, two groups. You have the sensory adaptive versus the under-responsive sensation seeking group. And you take these different measures. So this was efficiency and betweenness centrality. And what this algorithm will do is it will find a linear dividing line between them that keeps as many as possible of the under-responsivity sensation seeking on one side and the sensory adaptive on another. So in this case, this would have 100% accuracy, right? If this was your criteria that you use. Of course, we have 4 of these measures and we've got hundreds and hundreds of voxels that we're working with. So this is, again, it's in a very high dimensional space. This is just the simple description that we can give. And so what we do then is we'll get a result. So we'll figure out what the best classification we can do is across these groups. And then we have to create something to compare it to. So we have to figure out whether or not what we get is meaningful. So we do this by recreating this, but taking the labels on. So we take each individual and we scramble and give them a random assignment to a sensory phenotype group. And then we run it over and over and over and over again. And that gives us a null distribution. So it should be around chance. And then we want to look at our observed result. What we hope to see is that when we actually use the real labels that we do way better than we do if we just put random labels on them. So we did this, and the machine learning results were fairly good. So we were able to, so this does actually does a pairwise comparison. So we put in all the folks that were under responsive sensation seeking and sensory adaptive, and we did each pairwise comparison. So 50% here is going to be at chance level. This dashed line here is what we needed to hit for significance. And 7 of the 10 comparisons, we ended up being able to successfully categorize which sensory phenotype. So this is, again, remember, this is the what their parents reported their sensory issues were. A categorization based on that. We can tell that based on their brains, which is pretty cool. And so yeah, so we have 3 that are below the line here. This guy right here is the average. And when you look at this is the null distribution that we got, and this actually was our average. So we were at around a 57, 58% accuracy, which is highly statistically significant, but it's still not great. It's not something you'd want to take to the doctor's office yet, right? But what we were able to do with this is, so what we can do then is we can go in and we can, from that process, we can find the bits of information that contributed the most to this predictive ability. So this was overall including all voxels in the brain. Of course, there are going to be voxels. Probably most of the voxels in the brain are not going to be predictive, but which ones were really predictive? And we can look at this not just on where these were, but we can look at the network properties of that voxel. So is it that this is a region that if it's not properly connecting two different networks, so you have that like the betweenness centrality. If its betweenness centrality is really low, it's indicative of being in one particular phenotype or another. So we do this again, it's in a pairwise fashion. I'm not going to go through all of them because there would be 10 comparisons, 4 different, but I'm going to do the comparisons against the sensory adaptive groups. So, whoops. So you can get something like this. So each of these sides of brain, so this is the left side of the brain on the outside, the right side of the brain, and then this is the inside of the brain if you split it on the right down midline. So each one of these is a measure of one of these measures. So this is that betweenness centrality. So these regions here are regions that show differences between these two groups in terms of that between network connectivity that we see. So these are nodes, if they're different, they're more likely to, or if they have weak connectivity between different networks, you're more likely to be in one sensory phenotype or another. And you see some sort of things that you would expect. So you have, some visual regions that are indicative. Here you have some auditory regions. These are higher order auditory regions. In this gyrus right here is where primary auditory cortices are. But one thing that's really cool is you can actually see that. So these local efficiency and clustering coefficient, these were that tightness of the network. You can see that they're quite related to each other. So you see a similar pattern here, and then like here and here in these premotor regions here. But those are very different than what you see up in the measures of centrality, this betweenness centrality and strength. So this is, again, is how robust these areas are in terms of connecting the different networks. But again, these are similar to each other. So that's kind of a good sanity check, and you start to see the strength of this approach, where you can see not just where it is, but again, what network properties are really driving this difference between these two groups. And again, like we saw with those big matrices, oh, sorry, one thing before that. You can also look at this in terms of those 7 networks that I talked about. So, but in this particular comparison, we saw significant differences in both the ventral and the dorsal attentional networks between these two groups, and in the default mode network. So this is a much more much more fine-grained nuanced look, but you can also look at this in terms of the entire network. And just to show you what these differences look like across the sensory phenotypes, if you look at something, so this is the same group at the beginning, it's the sensory adaptive, but now we're comparing it to that low energy and weakness group. You can see that these patterns are quite different. So now you're seeing some primary auditory cortex. You're seeing a lot more differences, actually, but you also start to see differences. So these are in motor and premotor regions here, right here and here, which again, if you're talking about low energy, weakness, and movement issues. You also see the same thing down here with the local efficiency and clustering coefficient in terms of the motor regions. And the last example I'll give you is the taste smell sensitivity. So here you also importantly start to see things like olfactory bulb coming online, right? So this is what you're like your primary olfactory region. But even I wanted to point out two things. So I'm going to flick sort of back and forth between the previous ones. So just look at the differences as I go back and forth. Is we have some things like if you look at this area right here, some things where where it's just a matter of magnitude of difference. So we see a difference in terms of between this centrality and this temporal parietal junction. Regardless of what you're comparing it to, but it's just a change in strength, right? Whereas if you go to like the somatosensory regions here, it's a lack of a difference in one comparison. So there are some things that are not just quantitatively different, but qualitatively different in terms of the regions that are showing differences across these phenotypes and the type of information that they're processing. Okay, so that was all functional. Just checking the time again. I do need to move faster. That's just the functional. We also wanted to look at anatomical. So this is my graduate student. This is the portion that we literally finished up between yesterday and about 3:10 today. This was done by my graduate student Fan Yang, who was out this morning. This is literally him today, shoveling his snow. So we have three different neuroanatomical measures that we're using. We're going to use cortical surface area. So that's for each of these parcellated regions on the brain, it's literally just the surface area of it around that curved plane. Gray matter volume. So if you take the three-dimensional shape of that area, again, just the gray matter, not the white matter. What's the volume? And then also the thickness, so just the depth there. Now, if you've done basic geometry, you're going to know that these things tend to be related to each other, especially volume. So the first thing we did was we wanted to see whether or not we can reduce this information. So we ran a quick correlation. Each one of these rows is a different brain region, and this is how they correlated. Area and volume, it turns out, were really, really highly correlated with each other. So we just went with area and thickness, and we dropped volume out since it's so incredibly correlated. So the first thing we're going to do, so we had 76 regions that we looked at, and now we're going to have two measures, just surface area and cortical thickness. And we're going to run that's a similar, it's actually a little bit different, but a similar machine learning algorithm. And we're going to continuously add in bits of information and see whether or not we're able to predict which group they were in. And this, before I show you the results, so this is predicting this is not a pairwise comparison. So the baseline for this is going to be around 20% because there are 5 different sensory phenotypes. So using these two anatomical measures across the brain, Fan was able to predict with over 80% accuracy which sensory phenotype they were in, which in terms of predictive abilities, this is something now where you start to go like, okay, this can be very clinically meaningful. Which is really cool and exciting. So we wanted to do this, but we can do the same thing. So if you look at these different pieces of information, what you can do is you can withhold a bit of information. So you can withhold the gray matter thickness in region 1, right? And you can see how much that knocks down your predictive ability. And this can tell you what this tells you is whether or not that particular region is important for this predictive ability. So I'm going to show you, I just want you to look at the pattern here. Don't try and read the individual brain regions. There are 76 of them. You're not going to be able to read it. So again, each one of these is an individual brain region. This is the correlation coefficient. So these yellow ones that pop out are the ones that are highly predictive. And this value, it's too small to read, sorry, is going up to 0.4. So we're getting correlative values of 0.4. So some of them are quite significantly predictive. And that goes, but there are these like sort of islands. So this is, you can't see it when I point to my screen. This is the left half of the brain. This is the right half down here. But you can see that there are individual regions that stick out. I want to show you this because I'm going to use it as a comparison in a minute. We can also look at these areas of importance by actually plotting this in a heat map on the brain. And so these are the regions that we get. So we have on the left here, we have gray matter thickness, and on the right, we have cortical surface area. The brighter red a region is, the more significantly predictive it is. So in this case, we have huge swaths of temporal cortex into temporal parietal junction into the occipital, as well as areas in the frontal networks. And you see a similar thing. There's a bit of lateralization, which is I'm not completely sold on whether that's meaningful or not, where you have cortical surface area in the left temporal lobe and gray matter thickness in the right temporal lobe being a little bit more meaningful. Yeah, I'm not 100% sure. I bet if you plotted volume, those would even out. But yeah, so these are the regions that we get. Another important bit of information is sensitivity and selectivity of these. So what we have here, this is a confusion matrix. So this is essentially when you got it wrong, what did it fall out as, right? Sorry. So the true class on the y-axis here, these are what phenotypes the kids actually were. And then what we have in here is these rows is what they were, our model predicted them as. So if you look at the folks, for example, with generalized sensory differences, our model had 100% accuracy. If they had low energy and weakness, quite good. Then you get down, this is really interesting, I think. Then when you get down to the sensory adaptive group, so again, these are the kids that are sort of doing pretty well across the board. We did not do well. These guys were mischaracterized quite a bit. The taste and smell were all good, 96, as were the under responsivity, sensation seeking. But what also becomes interesting is if you look at, if you start from the prediction side, when these folks with sensory adaptive, when it was predicted that they had a sensory adaptive phenotype, those predictions were really good. It's just that anytime we predicted these other four, when we got it wrong, they almost exclusively fell into the sensory adaptive range, which to me suggests that the sensory adaptive phenotype itself has a good deal of heterogeneity in terms of its neuroanatomy, whereas these individual groups, the generalized sensory differences, the low energy weakness, taste, smell, and under responsivity, sensation seeking, have much more unique profiles in terms of their neuroanatomy. All right, last mini summary. Just wanted to add that yes, neuroanatomy differences can predict phenotypes. I'm going to rush through so we have time for questions at the end. But we also wanted to see whether they're specific to autism. And again, being conscious of time, I'm going to go through this quickly. Or are they transdiagnostics? So this is what we found with autism. We also have a large cohort of ADHD. ADHD, as many of you are probably aware, is highly comorbid with autism. We put them through the exact same barrage of analysis, except we haven't gotten to the functional yet. But when we do these phenotypes, if you look at so these are the phenotypes defined with only autism. If you do the phenotypes defined with only the ADHD group, you get basically the exact same thing. There are some differences. The movement difficulties don't have this like positive bump, for example, but you do see a group with taste smell differences, a group that's more under responsive sensation seeking, and then that sensory adaptive and general sensory differences. So this was none of the differences, even doing pairwise comparisons, none of the differences even had a medium effect size. So they're very similar to each other in terms of their phenotypes. We also wanted to look at whether they were related to the same clinical outcomes. So for these, I'm going to show you a series of graphs. They're the darker shaded bars, this green one, this red one, and so on. That's the autism group. That's the exact same data you saw before. These lighter shaded ones are going to be the phenotypes, but in ADHD. And what I want you to look for here is the patterns. So you see the patterns are almost identical across here. You look at the patterns across age, you see that same dip where the taste smell are younger and the low energy weakness group is a little bit older. You still see no difference in sex. The ADHD group has a little bit less of a sex bias, but so now I want you to look at the patterns here as opposed to the absolute magnitude. When we look at autism symptomatology, the autism group is going to show higher scores, But if you look at the pattern across the different groups where you see that, from sensory adaptive to generalized sensory differences, you see this big increase, a little bit down again with under responsivity, sensation seeking, and so on and so forth. The pattern that you see among these is nearly identical. It's completely reversed when you go to the ADHD scales, where the ADHD group obviously is scoring higher on these ADHD measures, but again, the pattern across the different sensory phenotypes is very consistent across diagnoses, as it is with the OCD traits. I also wanted to look at so this I think is interesting, and it harkens back to my first thought about that under responsivity sensation seeking group. Is that so what we have here is the percentage of each diagnostic group that fell into each one. So the autism group here was pretty even across the board, but the ADHD group had elevated levels both in the sensory adaptive, because sensory issues aren't as bad in ADHD generally speaking, but you also see a huge boost here in the under responsivity sensation seeking. So this is the one that I thought sort of seemed like me, but a bit ADHD, right? Then going into the anatomical components, so did they have similar neural issues? So this was the predictive ability that we were able to get with ASD. With ADHD, we got a very similar levels. It actually was a little bit more predictive earlier on, but our end result was about the same. And then if you look at areas of importance, again, don't worry so much about this, but if you remember back to the ASD, there were a bunch of like individual levels. So here, two things I want to point out. One is the scale here, the scale that we're working on. We're maxing out here at correlations of 0.1. It was 0.4 in the ASD group. So there are much fewer nodes of importance, and it's much more diffuse differences across the brain. So we're still able to predict with as good of accuracy, but these predictions are coming from a much broader area of the brain. And remember that scale when you see our little rotating brains here. So this is the autism group, and this is the ADHD group when we look at cortical thickness. So you can see that, again, with the exclusion of this laterality, which I'm not sure I necessarily believe yet, they look quite similar. But again, you have to remember that the scale here is different on them. And then if you look at oh, whoops, this is surface area, not cortical thickness. Sorry, I just copied and pasted the slide and didn't change it. And then last thing to look at, these were, if you remember, the confusion matrices that came up with the predictions in autism. We see almost the same thing with one difference. So again, the sensory adaptive group, but now we're also running into a similar problem here with the under responsivity sensation seeking, where we did, this one was confused in some cases for other ones. Again, we think because this is sort of a bit of a more canonical ADHD phenotype. So this one's accuracy was significantly lower in ADHD than it was in autism. Okay, I'm going to skip the mini summary. Okay, so next, what's on our to-do list of things that we want to look at after this? First, we need to get to the functional components of the ADHD group. It's just a matter of doing the analysis. We're also really interested to see if these phenotypes are stable across development. Do those taste and smell folks, do they actually grow out of it? Do people change as they grow up, or are most of them stable? And we also want to see how these different phenotypes relate to other outcome measures like employment and education and things like that. We're also planning, or we're in the process of running a study with Spark on whether or not there's actually a genetic component to the phenotypes, whether they're heritable. And then maybe I can convince Rob to do some looks to see pre and post if pharmacological interventions actually change how these phenotypes look. So last slide. Take-home points. So sensory issues are heterogeneous, but they're not necessarily unique. So even though a one-size-fits-all approach to them in terms of providing supports won't necessarily work, we might be able to have a small number of different sort of like groups of remediations that we just have to find the right one of those small ones, not necessarily reinvent the wheel with each kid. It's also, I think, really important to keep in mind that these sensory differences have cascading impacts and can impact other clinical symptomatology. That what things that may present as a more high-level issue may actually really have its origin in sensory issues, or at least an influence of sensory issues. And that can be found not just in identifying where things come from, but also finding targets for support. And then last, that these neurobiological differences, that there are this is not just behavior, it's in the brain. There are differences in the brain, and that these neural differences may be able to inform how we are going to target our interventions, particularly for pharmacological therapeutics and things like that. So with that, I am out of time. So just want to thank all of our funders and all the folks in the lab that have been working on this. It's always odd doing the webinars when you can't see anybody's face. Hey there, Rob. Jason, are you there?
Yeah, I'm here.
Okay.
Like you might have froze.
Okay. Oh, what is my oh, never mind, that's from Rob. Rob, I'm an associate professor. So Jason, do you do the
Yeah, so we can continue with questions. So there's a little
questions?
Q&A button you'll see maybe at
Ah,
the top or bottom of your screen, depending on your setup. If you click that, you should be able to see somebody's asked. Jessica has asked a question.
right. I am
I can put it
Q&A,
too.
yes.
Okay.
Okay. Do you believe behavioral? Um, so the question is, will behavioral interventions have the potential to modify these phenotypes versus purely pharmaceutical? I think in the long term, I would say yes. I think especially with that potential finding. Again, it's not longitudinal, but the fact that the taste smell group was younger kind of to me indicates that they grew out of it because these were uh, striated across, they were pretty evenly distributed across different age ranges. Oh, it's Jessica. Jessica, hi. I, and so I would think that behavioral interventions could change, but I think the other thing to think about is you can change which behavioral intervention you use based on their sensory phenotypes. I think that's actually where I would say I think that's less speculative, I guess, if that makes sense. Ben Goldberg, how many children had treatment for their sensory? I am not sure who if any of them had been receiving treatment for their sensory information. I'm not sure if that's Rob, do you know if that's in the pond
I,
data?
I, I doubt it because I think as far as I know, and I'd have to check, I think the only thing we keep track of is, frankly, on a going forward basis would be medication treatments for people and
Hmm.
whether they've changed. And I mean, it's a great question that Dr. Goldberg has because, first of all, it'd be interesting to know if again, if there's a way to do sort of pre and post, do things actually change over
Hmm.
time? And I don't think we keep that data actually. I can't think
Yeah.
of what it would be.
I mean, the nice thing is, so Pond, I mean, for the non-Robs, so Pond is starting its second wave of collection. So we are going to be getting longitudinal data on these kiddos. So we will be able to actually see it. We just, have to wait for the data to roll in.
But and I think also, Ryan, with your statement, question, comment, we'll say about treatment before and after. So you and I can talk after. We're starting another clinical trial shortly of for anxiety stuff, and so we can look. If you're interested, we
Hmm.
can do sort of before and after kinds of measures. And I've talked, I know, with you and Julio loosely about stimulants and whether
Hmm.
some of Julio's data would suggest
Yeah.
they help. And I've done things with a couple of patients, been really equivocal, sort of not necessarily in a research way, just clinically to see if it made a difference. But it's something I would like to go back to at some point and revisit. I
Hmm.
had a couple of questions, Ryan, about your data. So first of all, you think with the purely, first of all, what is do we know, and I know I've asked this before, but the relationship between any sort of objective measure and the short sensory profile, is that known?
Objective, but you mean like a psychophysics measure or things
Whether
like that?
TMS or anything, yes.
Yeah. So oddly, so the short sensory profile itself, especially as a parent report measure, can only really look at sensory responsivity. Because what you're reporting is how the kids respond to it, right? I mean, a parent can't look under the hood and see what's going on in their kid's brain. So we have done a series of studies. I think I might have actually presented these at this rounds, I don't know, 7 years ago, 6 years ago, um, where if you do measures of sensory, what often gets called sensory sensitivity, but really it's reactivity again, because it's a parental report. And you do psychophysics measures of, like we vary, we put a Gabor, a sinusoidal
Yep.
grating on the screen, looks like a potato chip, right? Embedded in noise. And you make it dimmer and dimmer, and you see exactly how bright the stimulus has to be, right? And so that's a measure of like pure sensory sensitivity. How bright does something have to be before you can detect it? Surprisingly, the correlation was almost zero. And actually, I think we got a fund from Dr. Goldberg to do that
Yep.
research. Um, it was almost zero. So they were measuring very different constructs, but both of them were still highly predictive of autism severity. So
Hmm.
I think they're different, and you can conceptualize getting to that, or like let's say, oh, call it a reaction. So if you think of a kid in the bathroom, right? The big thing is that the hand dryers, right, in bathrooms
Yep.
drives autistic kids crazy. Um, if that goes off and you cover their, and it sounds the same as it does to you and I, and they have this response where they cover their ears and things like that, that would be, I would classify as over responsivity,
Right.
right? If they're perceiving it the same. But if it sounds like a fire alarm going off right next to their head, you should put your, like that is more of like sensory sensitivity, but what you would see on the outside is the same thing. You see the kid.
Hmm.
And so we've started to do some EEG studies looking to see exactly where these differences come out, but it does seem to be that they're different constructs. We don't know exactly where in the processing pipeline these are, but it's actually a fairly important question, because if you think about how you would treat those two things, um, if it actually sounds like a fire alarm's going, you're not going to want to do something like exposure therapy. Then you're just torturing the kid,
Yep.
right? But if it's something like if it's
But
just
I think we use the
if
term
they
what is it? The enhanced interrogation, I believe is the term, not
yeah,
torture.
yeah.
Thank you.
But if it's something where they hear it the same, and they're just, it's the response that you want to do, then you could use something like exposure therapy, right? So get that, I think that can actually have good clinical importance to it. So
And I think also potentially ideological, because I wonder, and I don't want to oversimplify it, but I also wonder, particularly with the noise stuff, how much of that's just related to an exaggerated, or not just, but partially an exaggerated startle response. Because I've seen a fair number of kids over the years now clinically who will be getting on the elevator and they may be a bit uncomfortable and will actually cover their ears getting onto the elevator, but elevators don't generally make noise or not
yeah,
significant noise. And so I don't, and I've asked their parents, what's that about? Like, why do you think they're doing that given that there's no noise? And these parents have generally said they're not sure that they've noticed it, but don't know how to make any sense of that.
yeah,
And I wonder if that's about anxiety or predictable. I'm not sure.
yeah. Or just that it's the one thing that they actually have control over, so they're
Yep.
going to reduce what they can. Yeah.
So the other thing, so you had said, I think with the phenotypes, or the clusters based upon the sort of sensory profile, I think you used the word predictable, that they were predictable phenotypes.
Hmm.
What does predictable mean?
Um, so what I mean by predictable might not be the best word, but I mean is that they're not that they actually, you can predict what goes together. So you can predict one,
Ah,
if you
okay.
have one big issue. So like they're reliable might be a better word.
Yep.
That this pattern is seen across a lot of kids.
Okay. Fair.
It's not that it's just random
Gotcha.
assign.
Yep, yep, that's fine. So, are there questions for Dr. Stevenson? We can pick his brain while we've got him here if people have clinical questions. Also, that Ryan is one of the few pure research people that I know of who actually has qualified to administer the ADOS and things like that. So obviously, he's gone out of his way to sort of look at the clinical entity in addition to just the purely research aspect of things. So, right, are you surprised that that cognitive ability did not seem to relate to the severity of the
Yeah. Yeah.
problem?
No, I mean, Yes, I was. So especially when I, with the generalized sensory differences group, I went into it with the idea, once I saw what the phenotypes were, because that was more exploratory, I assumed that they would be more severe across the board on all of the measures that we looked at, which ended up not being the case. But at the same time, I think that's actually quite interesting and telling because it's not, it's to me, it's more useful if it's just more severe autism or more severe disruption. That's it's not very specific, right? It's not something that you can really use. But if it's actually that they're showing the same average cognitive ability, they seem to be doing okay in this area, but not that area. They're not doing that, that's something that is to me is more indicative of something that, yeah, that's more meaningful in terms of sensory processing. Because then it's not that it's the sensory processing, it's just that sensory processing is one of the everything
Yeah, their
that they're
brain just
not
doesn't
doing well.
is worse, so to speak.
Yeah, everything sensory is just one other thing that they're showing more issues with, but it does seem to be that they're more patterned than that.
But because I think one of the issues we have in Pond is there aren't necessarily a lot of people with significant cognitive impairments who are in the sample size. So did you
Mhm.
include people with intellectual disabilities in that group?
We included everybody.
Okay.
Yep.
So
The
that wouldn't
average
account for it.
was, I can go back to the side, I believe the average was about 80 in
Okay.
terms of IQ.
So that
But
doesn't explain why there wouldn't have been that finding, though.
no, yeah, we tried. The only reason we excluded the 80 the comorbid autism and ADHD is so we could make that comparison
Right.
between them.
So if there are other questions, we have Dr. Stevenson here to say, but so right, if the DSM, I think one of the problems is the way that the sensory criteria is written is there are three almost contradictory features, right? So you can have people who are overly responsive or perhaps preoccupied with sensory things like looking out of the corner of their eyes and stuff,
Mhm.
or people who will cover their ears and stuff, but also they include under responsivity. So people who
Yeah,
don't seem to respond. And so there are three I don't know if they're mutually exclusive, but they're sort of I don't even know if they're independent, but they're very different. So if you could
yeah.
rewrite, if the people at the American Psychiatric Association came to you and said, Dr. Stevenson, we would like you to rewrite the DSM criteria for the sensory stuff, what would you say?
Well, so that is not something that I would take issue with.
Yeah.
So this is not answering the exact question you have, but the way that you're phrasing it there is that there are people that are over responsivity and other people that are under responsive. That is where I would say that I wouldn't agree with that logic. So it's more of a so you can have one individual. So if you take this short sensory profile, you actually get a measure of over responsivity and under responsivity. And the two are correlated with each other, not anti-correlated, but directly correlated with each other. And a lot of it is contextual. So I have a clinical student, Michelle Lazowski. Actually, I'm never mind. Who's doing a study. She's pinging people with text messages throughout the day to get actual, like real world in the moment, in their home environment, when they're out and things like that, and asking about these questions. Or asking them these questions. And individuals, based on the context and what's going on, can be either hypersensitive or hyposensitive. So an easy sort of, if there are any like educators or in the room is, you might have a kid who will walk through the classroom and bang his thighs on the desk to the point that he gets bruises but doesn't notice it. That same kid in a different context when it's not expected, you might walk up to him and tap him on the shoulder and he jumps, right? So one would be oversensitivity, one would be undersensitivity or under responsivity, right? So a lot of it is context dependent. So their sensory issues, they tended to be a lot more hypersensitive when they were out, when they were with other people. But it was really cool when we took them. Yeah, when she got outside of the lab and actually um, was doing this research when they're in different environments, you get very different levels. And actually, one of the most important ones was the predictability of the situation. So like your ability to control it. So when they were in control of things, if they're the ones picking the music and they might want to crank the music up. I mean, this is a simple
Yep.
example, but
Yep.
but if it's something that's out of their control, it's way more distressing to them.
But that's partly why I wonder about startle and stuff like that and how much that
Yeah.
might, it's not startle because if you're looking at somebody, they're turning on the vacuum cleaner, presumably you know that it's being turned on.
Yeah.
But
Yeah.
you're right, that's apparently in the car, their kids will put up music as loud as they want, and it's not a problem. But I think
Yeah.
you and I have also talked about, is it the pitch of the tone or what, there's something different.
Yeah,
But I
there are
think
lots of
including the whole thing about people who look out of the corner of their eyes and call it that sort of over interest, I guess, in sensory stuff. It just seems to me that, I mean, that's interesting what you're saying about the correlational stuff and the context. I don't know how you operationally define that, and maybe nothing is really operationally well defined in the
yeah.
game.
Yeah, well, the way that she defined it was they were asked about, like, of course, like what their sensory experiences were, but they asked things like who were you with, like who's with them at the time, what was their location, like were they at school, were they at work, were they,
Hmm.
like got all of the different environmental factors um, into it, and then like measured. Also asked, were there any positive like positive valence measures of it, whether or not they had control over it. Then like antecedents also, like were you hungry, were you in a good mood, were you, that level of stuff. But yeah, the context around it really changed. So I think in going back to your original question, I don't think that it's that a person is either hypersensitive or hyposensitive. They can be
Yep.
both.
But I think that's really important. Just think about sort of future research and paradigms that we use, that it's not necessarily one or the other. And maybe we need to not, it's not just do people over respond, but in what context do they over respond
Exactly.
and stuff like that, because it's obviously not an all or none
Yeah.
phenomenon from
Yeah.
what you're saying.
Well, and you can't base everything on what you have when they're in this weird lab environment that they've never seen
Yep.
before with somebody standing over. So yeah.
No, that's really fascinating, actually, right? So all right, if there are any other questions, now is the time to ask them. I think so. Jason Wittes just made a point that thank you again, everybody, for joining us. So there is a link in the chat for qualification people could fill out an evaluation only with perfect marks for Dr. Stevenson, please. But also that really helps us. And if people have suggestions for future speakers, that's really useful as well. So on February the 8th, I believe, is it February the 8th, Jason?
12th.
Well, sorry, I was just testing it. Allison Cox, who's a BCBA at Brock University, is going to be talking. Our next speaker is going to talk about the relationship between psychiatry and behavior analysis, which is a really interesting topic. And she's developed or is developing tools to sort of use medication and monitor how that's helping with behavior analysis, which I think is a really neat idea. So we look forward to that. And again, Ryan, I just want to say thank you for doing this yet again. You've been
Yeah,
a
of
huge
course.
help, and the talk, as always, was fantastic.
Thanks, Rob.
Yep, and we'll be in touch. Say hi to Magali for us.
All
Yep,
right, I will.
yep.
All
Bye-bye.
right. See ya.